Up next, Antton Alberdi, who is an associate professor here at the center, has kindly volunteered to host a dialogue between not only Thomas, but another of the big names in holobiont work, Mónica Medina, professor at Penn State University. So big thanks to Mónica for agreeing to do this with Antton. So welcome back, people. Antton will explain more, but would you like to come forward? I'll get off stage.
Welcome everyone. Let me get my notes first. So now we are going really holo, from single Thomas to holo Thomas. So now we are going to extend a little bit the scope. Also, we will take as a baseline some of what Thomas has told us in the last hour. Before that, though, I will explain you what is the intro story of this symbiotic dialogue. And for that, I will need the pointer. So 2 years ago, as Thomas mentioned, we organized this, or Tom mentioned, it's also Thomas. We organized this first Applied Hologenomics Conference, and part of it, we organized the symbiotic dialogue, the first symbiotic dialogue. And we want this second symbiotic dialogue to be the next step. We would like to elongate that chain. At that time, it was, you can see myself here, and it was Seth Bordenstein, who is there in the audience, and also Henna Kailanto, that were the two invited speakers that for 1 whole hour, they expressed their ideas, their opinions, their experiences about talking about 4 main concepts that I will try to remember. I have notes for that, which were symbiosis. Those are the new ones. Wait a second. There were 4. There should be somewhere. Today, we are going to take the last one of those concepts, which is symbiosis, as the first one, as the first concept. And then we will continue. We will navigate through 3 other concepts, which are complexity, ecosystem, and challenges. And we will navigate through this using those concepts as the starting point, trying to cover different aspects around the very broad topics around this concept. Before we go into this, though, I would like to explain you how we are going to do to be able to interact with you. So I was told that it would be too chaotic to be running with microphones all the time. So what we have done is to prepare this Slido page, a Slido question and answer audience, that you should be able to get in using this QR code. So if you use your cell phone and you zoom in and then get into the website using this QR code, you should access to something that looks like this. And here you will find that there is this type your question field where you can use either for typing your question, if you have any question you would like me to ask them while we are talking about different topics, I will try to include those questions. We have some idea of what type of topics we would like to cover, so I will try to make the decision on when to ask those questions. But we would also like to get your comments. So if you have any other comments, you would like to say something, you would like to interact with them, you should be able to do the same. Also, write in your comment in this type your question field. Once all these questions and comments are in the system, all of you that connect to the question and answer session will be able to give like to those questions, and that's going to be extremely important for me because you will do my work basically, and I will have less work to multitask by asking questions, responding, and then filtering your questions and comments. So feel free to like all these questions and comments that you like the most, and then I will do my best to include them into the discussion. And please, as we have a really nice, cozy, friendly environment here, use your name if you want, but we would really like to get to know who you are when you are giving your opinion, asking your questions, and then I will spell out and say who is the one that is questioning or adding a comment. So, without further ado, we will start with the conversation. And before I ask you the first question, so Thomas has already introduced himself by this really great presentation for the first hour. But now we have next to Thomas Mónica Medina, who is a professor at Penn State in the US. She's an organismal biologist and studying mainly marine biology, so marine holobionts, with a special focus on cnidarians. So that's the very short intro, and I'm sure that you will have the possibility now to tell the audience much more about what you do. So now that we have everything ready to go, and I can see that Slido is ready and should be working. So we will start with the first concept, which is symbiosis. And for symbiosis, for illustrating this concept of symbiosis, I have selected this artwork by Shoshana Dubinier from 2012, which is called endosymbiosis, actually. And in the previous talk, Thomas, you have talked about this process that occurred millions of years ago, and you also touched base on Lynn Margulis's work. And what you have not mentioned was that, or you kind of said that there was some issues with it, and then there was some disputes about it, but it took some years until this idea was really sunk, and the community really accepted that was the case. For those who have not been involved with all this thinking around the holobiont idea, hologenomes, etc., I would say that something similar probably have happened in the last 15 years, in the first 10 years at least, since these ideas were first released, and there were some disputes. Do you see any kind of resemblances? And did we learn anything from those disputes that happened 50 years ago for the conversations, the disputes, the discussions that have happened in the last 15 years? I will start with you, Thomas, and then we'll continue with you, Monica.
Thank you very much. First of all, thank you for setting that up here, and this makes only sense if you are also part of us here. So we are here in one room and sharing ideas or thinking about certain issues. And if Antton asked what is similar or different to Lynn Margulis's key statement that the origin of the eukaryotic cell is actually a symbiotic event, which happened because a cell was engulfing a bacterium, which then became a mitochondrium, or in the plant cell, a chloroplast, then of course, don't expect, and this for all of you, if you make a groundbreaking, something completely novel discovery, don't expect that you get greeted by the community with enthusiasm. You get highly skeptical comments, and so this idea took a long time to really get accepted. But it's now, I mean, every school kid learns now the endosymbiotic theory, or the endosymbiosis, the origin of the eukaryotic cell and the eukaryotic life. It's getting more complicated these days when we talk about hologenomes, and all of you who are in the field here and who are pioneers in the field, they have realized and have experienced that, including myself, that if you now come up with your strong evidence and belief and convinced that organisms are multi-organismic entities, and if you separate them into their single parts, they are not fit in terms of evolutionary terms anymore. That also is not really, the first reaction is not enthusiasm, I can tell you, from the community, but highly skepticism. And because, of course, it's getting so more complicated now, and particularly because it touches also evolutionary, much stronger evolutionary issues than Lynn Margulis has touched. And at a very early time, and Seth Bodenstein was part of that discussion. And, you know, I would say friendly conservative evolutionary biologists, I mean, the whole genome theory of evolution doesn't mean, and Eugen Rosenberg and Ilana have not put it out to make a new theory of evolution. It just, they have made a new point that if you think about evolutionary processes, we have to think them in a multi-organismic context. But that then came the question of where is then the level of selection, and et cetera, et cetera. I mean, I don't think that we need a new theory of evolution, but indeed, everything is now much more complex, and particularly, and I give then to Monica, the crucial issue of adaptation. And I mean, how can organisms quickly change to a changed environment? And that is a fascinating but not understood process. And Eugen Rosenberg came up with some original papers before his hologenome theory of evolution, where he proposed that indeed the acquisition or the getting rid of certain microbes may change the fitness and the adaptability to a changed environment. So there's so many aspects now, and we have to really follow them up on a mechanistic level. We have to understand what are the selective processes which are working there. Don't get disappointed if you don't get enthusiasm. It is a time of, it just tells you you are a pioneer in the field, and pioneers are having sometimes heavy weather and heavy water. You have to go through that. But if the experiments are clean and causality driven and mechanistically driven, at the end you will be right. And Lynn Margulis was right, Monica.
Thank you. Well, it's great to be here. It's the first time that I participate in a panel like this, so I feel like I'm a bit in a therapy session. So bear with me. But so along those lines, I think I have two things to say. One, that I think context, sociological context, has a lot to do with how scientific growth, like knowledge, evolves, and your experience and your interest, of course. So I did a postdoc in a Proteus lab, Proteus evolution lab. So Lynn Margulis was our icon, you know. So I always embraced symbiosis. And then I started working on corals, and we haven't talked about Forrest, but Forrest Rower is a coral biologist, mostly microbiologist and virologist, that coined the term recently for us, and I've asked him, I said, did you know Lynn came up with this term before you? And he's like, I think that's a case of convergent evolution, you know? And he just found it useful, and maybe he's dug deeper now, because I know he's working with philosophers of science on what happened with the term and how multiple people have used it, because the photosymbiosis people have used the holosymbiont concept to just refer to the coral and the algal symbiont. And it didn't stick, you know? But when Forrest came up with the holobiont concept for corals, we all started using it. And then I started to read, you know, Seth's work and the pushback, and I was like, wow, those insect people, oh gosh, you know? Like, they were very unfriendly, and they were very prominent evolutionary biologists. But then I started to see your work and Scott Gilbert's work, you know, who wrote the textbooks in development, and I was always, you know, an Evo-Devo scientist wannabe, so I follow the literature, but never from afar. And then I started to see how the developmental geneticists started to get into microbial interaction research, and I thought that was a new revolution for Evo-Devo research, because they started to, you know, Margaret and you, and I think that's sort of when Seth and I met, and then Seth's like, yeah, it's been rough, and I'm like, really? Because that wasn't my experience, and he's like, yeah, you marine people have had it easy. So that's what I refer to sociological context, you know, that for me, I never felt that aggressive pushback, but I saw it, and I was just like, ouch. And I've felt it from other places, you know. Coral people are not the friendliest always, you know, and I'll tell you more about that, but when I talk, but it's, you know, I think it's sort of where you are in your scientific quest and who you're surrounded with. But I feel that coming to present time, there is a lot of things that are happening, you know, the center here in Copehagen is revolutionary, is led by people that are coming at it from very different angles and different perspectives, spatial temporally, ecological and evolutionary questions, and even all time, right? So it's a reputable center with great funding, and then all these senior scientists from different disciplines embracing it, and then the climate crisis. And if we are going to help address this climate crisis, I think microbes is where it's at. There's no time, especially for corals, there's just no time. So, it's just take it or leave it at this point. So I think that, to get back to your question, I think that there has been multiple series of debates intellectually that have shied away from practical and applied issues, and we can no longer afford to do that. So I think we're at a huge turning point, and meetings like these are really important because are bringing basic fundamental science with all these deep knowledge in evolutionary and ecological theory, and Joan is going to tell us about that this week, and I'm so grateful that you're here and part of this community because you bring so much standing, to what we are all trying to do. So I think that the bad days are behind, or I want to believe, because I need to find optimism somewhere anyways, so I'm choosing to find it here.
If I can just add up one word, which is in support of what Mónica said, I think the conference here is called Applied Hologenomics. And I talked to Seth briefly when I came here before this afternoon. That means something, thinking about holobiont and hologenomes is useful and can change. And as soon as we have entered that, and we have entered that, then as Mónica says, all these theoretical issues, yes, they may still be interesting to think about, but if animals can be more healthy, made more healthy, or plants can be made more healthy by thinking about the microbiome, etc., and medical issues can be understood in a more holistic manner and better, and patients can suffer less by using this hologenomic thinking. That's it. We don't need more, and this is then your challenge and your task. If you bring that up, and if you bring it up in startup companies, or if you bring it up in papers, I mean, there is no argument anymore that this is not true. It is true, and it even helps. And then all this discussion is over. Yeah, thank you. Yeah.
I actually have a strong piece of evidence that shows that. There is a Maserati parked outside there. So someone here in the audience came in a Maserati, apparently. So it means that, yeah, apparently we are not a niche field anymore. So let's continue now, and let's transition to the next concept. And this will be related to what you said, Thomas, also in your talk, that it's not about one microbe. It seldom is about one microbe. It's about communities of microbes. It is much more complex than just one-to-one interactions, usually. And in that sense, we have illustrated that with this artwork by Gustav Klimt from 120 years ago almost, which is called the Tree of Life. And what I would like to hear from you is, and this question is mainly for you, Mónica, because Thomas has given us his impression of why to use, why is it valid, why is it useful to use hydras as models. You chose jellyfish, mostly for your work. Could you let us know how complexity plays a role in this interaction between jellyfish and their associated gut microbes, and what is the kind of information you can get from those interactions that transcend the jellyfish and the jellyfish holobiont in general?
I was raised as an evolutionary biologist. I failed as a population geneticist, but I was okay as a phylogeneticist. And I came into genomics at a time, I joined a genome center that had just finished human, and they didn't know what to do with these sequencers. And they're like, oh, there's evolutionary biologists walking around. So all my protist knowledge came in handy because I knew all these marine protists and stuff like that. So I started to really look at genomics from a tree thinking perspective, which was very new when you have only spent 20 years sequencing human and nothing else, right? So I think that gave me an edge to think comparatively in terms of comparative genomics. But I was raised also as an invertebrate zoologist, and I was always interested in invertebrates and symbiosis and understanding early branching lineages, as Thomas already showed, is very helpful in understanding evolution. So cnidarians are basal animals. And I initially started, and I'm mostly going to be telling you about corals actually this week, but working with corals is really challenging because they're endangered and it's hard. The biology is hard, the working in the field is hard, permitting is hard, and it's so complex. It's very different to look at an embryo and to look at, a larva and a cell polyp, and then look at a colony that is 400 years old, and the microbial assemblage there. It's really tricky. So listening to what the people working on model systems could do was just so inspiring. And I'm like, okay, well, we need to try to find the closest thing that does the same thing or similar things. So working on the jellyfish has presented its challenges, but also the opportunities, because we can look at the establishment and the assembly dynamics of the photosymbiosis, but then all the microbes that come with that. And it's giving me a way to partition that complexity more easily. And for us, it's early days. Hydra is, a century in terms of, right? And the jellyfish, some people, including Thomas, have started to work on another jellyfish, but it's not photosymbiotic. So this work that we've done is very, very recent. But we have been able to make big headway into understanding the assembly dynamics of photosymbiotic organisms using a model system that can apply to many photosymbiotic organisms across the tree of life, including many other animals, because they all establish symbiosis with, or not all, but many, with the same algal symbionts that come with their own microbiomes. So I think by focusing on a particular model system, then you can take from that and try to understand all these different branches of the tree that present similar life history patterns to come up with universal ideas of how photosymbiosis and photosymbiotic holobionts evolve, because there are protists that are photosymbiotic with the same endosymbionts. They are animals. They're, primarily protists and animals. I don't think that there is other photosymbiosis of that nature, but they're pretty prevalent because they're very successful in the tree of life. So I think, I don't know if I have answered your question, but that's my answer.
Yeah, thanks. I brought here an example, a very iconic example of this alluvial fishery and the bobtail squid light organ interaction, in which it's been very iconic because it is really beautiful first, and then it's quite simple in the sense that we have an organ that gets colonized by one specific bacteria, and this interaction has been understood now, has been studied for many years, and we can know a lot of the nitty-gritty details of how this interaction happens. And Thomas, you in your talk also mentioned a lot of the mechanistic connections that happen between the hydra and the different bacteria, and what are the actual gateways and the ways they can communicate, and so on and so forth. But this is a very simple example in the sense that it is almost 1-1 in the skin light organs. But if we go to, as humans or vertebrates in general, suddenly we have interactions that is 1 to thousands and thousands. And you mentioned about the mechanistic interactions, and you have also mentioned the word models many times. How close do you believe we are to have actual mechanistic models that can allow us to predict how these interactions will happen in different conditions, and how this is going to really have an impact on how the holobiont will adapt to an environment, what you said, or to even evolve in the future when the conditions are changing.
Before I answer this question, I go back one step, and particularly for the younger generation, to complexity. I was raised in a fantastic lab, and I was a fantastic mentor with one mission in the lab: avoid complexity. And so make your system as simple as possible. One cell cultured under defined culture conditions, and try to understand that is even already for this generation of experts super complex. And I remind you that the origin of molecular biology was happened in LA in the lab of Max Delbrück. They used phages because they thought that they are the most simple version of life on Earth, where one can understand the fundamental rules of life. Today, I ask you, embrace complexity. Do not lose the power of reductionistic approaches, but we have to accept complexity. And we have the technological means to do that without losing depth. That's always the argument for reductionistic approaches. So but if you avoid complexity, you will never reach the core of your question and your problem, thinking about complex diseases, which are rising and exploding these days. These are all complex between microbes, thousands of microbes, and host tissues and environmental factors. But you have to see that holistically. And I'm always optimistic, I think, as Mónica, and I think a scientist who is not optimistic, I don't know many of them. But I think today, with these fantastic new technologies, and I know that Tom Gilbert has organoids in his here in his center. I mean, we can reduce complexity to some extent without losing mechanistic insight. And the other thing which I really want to tell you is you have to stay at a mechanistic level and causality driven. All correlations. There is, the abundance or the disappearance of this is correlated with that. We know that since 10 years. This is, forget it. Show if there is a causal interaction and what are the mechanisms and the biochemical language, then you make a contribution. But you can do that. And models, there are simple models, and simple models have their limitations. Of course, a hydra is not a human, and a jellyfish is not a human. And I know too many medical persons who says, who cares about invertebrates? They never will model the human body. On the other side, I'm invited by gastrointestinology societies to talk to them because they realize that, the mechanistic and causality in humans is not so easy. I think organoids are a fantastic way of how to somehow get around that problem. And organoids is also an interesting issue because they were the worst thing what can happen to an organoid culture is if it gets contaminated with bacteria. And you learned that in the early days of cell biology. But now you can controlled add microbes, and then you have your readout system, and you can see what happens there, and you can add a less or more complex a mixture of microbes. So this is just a bit challenging, but science, that's the fun part, and I see no reason and no limitation.
Mm-hmm. We have actually one question from a mysterious Holotom, who knows who this guy is, that links this discussion about the complexity with the previous conversation we had about thinking the idea and so on. And Tom is asking whether do you think that the pushback we face on Holobiont ideas, or we used to face, is due to lack of belief in the idea itself, or is it because they don't believe that we can actually study it in a meaningful way, which would be what we are talking about now. Mónica, do you have any?
Can I repeat myself and say, as soon as you have proven that it's useful and that you can treat a certain disease to some extent, or you can make an animal better, more useful, healthier, whatever, or a plant, by interfering with the host microbiome interaction, it takes away all the discussion. That's it. And so I think, yeah. Monica.
I also think, am I losing this? Can you
It's
guys
okay.
hear me?
No, yeah.
Okay. Going back to the historical nature of disciplines, especially evolutionary biology, evolutionary theory was developed without data, following, historical events in the West primarily. So, alleles were thought of without we knew what the gene was. So I think there's been I'm talking semi-naively here, so please chime in if you think I'm wrong, but I feel that, a lot of the theory was developed using a Mendelian genetic selectionist approach, and now we're inundated by data that don't always fit the Mendelian population genetics approach, and that requires new ways of thinking and approaching large data and evolutionary theory, not only from the modern synthesis perspective, but from a broader synthesis, inviting systems biologists and all these people that think differently to deal with large data. And that's not something that the field grew with, the field grew without data. I remember the 66, was it Lewontin? Oh, 2 protein isoforms. Wow, we have 2 alleles. And now we have genomes that have all these SNP data and transposable elements and microbiome genomes that are interacting at different times of the life cycle of an organism, having different selective pressures across life cycle. Last year we had at Penn State, help me, Seth, the evolutionary medicine, what's his name? Who was, he was in Switzerland and then at Yale. Yes, and he was arguing that we are messing with the system so much that we are selecting for different fitness advantages across a very different lifespan than we were used to. So we really need to modify how we think, even with these traditional modern synthesis and Mendelian genetics perspectives of how we look at fitness. And if we throw in selection on the microbes that are putting selective pressures on the host, at different times with different selective values, then it gets really complicated fast, and that requires new ways of thinking and approaching the issues that are either non-existent or in their infancy and that are going to get a lot of pushback. And that's what happened to the Rosenbergs, because they were not theoreticians and they're bringing all these modern synthesis ideas and retweaking them, and all these theoretical population biologists were pushing back really, really hard. And I think we're past that a little bit, and I think we as a community are becoming more organized and trying to put out maybe controversial ideas that require new theory, but I think that is really, really necessary, because now we have a super data-rich field with theory lagging behind, which was the opposite of what was happening in the beginning of the 20th century. Look at Kimura, right? He had to struggle. He was a genius. And he's like, what is this dude saying about neutral theory? And it took him decades to push back on the selectionist perspective, right? So now it's not a single gene, 2 allele kind of situation, but whole genomes and hologenomes. Of course, it's going to be challenging. I don't know if I'm answering the question. Okay. Just went on another rant there, but I hope it's helpful.
No, let me share with you one experience, quite a disappointing experience of mine. I have applied for 3 times for an ERC grant, 3 times for actually addressing adaptation of vertebrates aided by microbiomes. And the first one, the main criticism was that, you don't have enough evidence to show that this is meaningful enough. The second one was another excuse. Then the third one, when I came up with an actual preliminary data and everything had its preliminary data, I say, okay, that question is already addressed, so it's not meaningful anymore. So it felt to me that it was not that much about the ways presenting, but it was more about the perception of the evaluators. Mainly, this was LS8, this is traditional ecology and evolution mainly, that do not really buy the idea. What would be your strategy? In this case, for me, what should I do next time?
Keep going.
Keep going.
Keep going. Does it work?
Yeah.
Keep going. This is for sure. Somehow I did something wrong. I'm not sure. I cannot shout anymore. I shouted all with the soccer ball. But keep going. Of course,
Yeah, something is wrong with the.
I think it's muted. Muted. Yes. Oops. Yeah.
Here we are.
Okay. Thank you so much. See, we talk about hologenomics and cannot turn on this simple advice here. Okay, that's great. This is a normal reaction, and I think, keep going, and you believe in your data, and as you have to prove, and there is no other alternative. You can only convince by facts, and then you need maybe a bit more. You don't get the big money, but maybe you get the smaller money, and do with the small money a case study, and in applied hologenomics or whatever, and show then that yes, it worked, and. Step by step, this is this infamous statement that one funeral at a time, when you make a new discovery, that's it needed to get accepted. But I think that's not true. I think you really can make your point, but I'm not surprised about what you realize in your grant or your papers. I mean, this is you're not alone.
Thank you. Thank you. That's
Can I chime in
yeah,
there?
sure.
I can so relate. I tend to think outside the box, and I'm very different, and I've late in life really have come to embrace that because it's hard otherwise. But I think to encourage the younger people, I'm still standing despite all these misadventures. So it's not easy, but I think as you grow your network of people, and it's something that I did not do. I didn't have, this kind of open discussion or mentorship. Tap on your network, like here there is 220 plus 60 people that believe in the holobiome concept. So talk to those people and cultivate your reviewers, reviewers and mentors and sponsors, and just say, I'm struggling with this and this and this and this, and somebody will have that new perspective of maybe pitch it to this way or to this funding agency, and the senior mentors be like, put me down as a reviewer. People do it all the time, So if you see somebody you haven't collaborated with in this group that is friendly towards your idea, put them down. You may want to ask them or not, but just do that, because I learned the hard way, and it's still hard. A female woman of color in science, are you kidding me? But I'm still here from a country, not Switzerland. I'm from Colombia. So, it has not been easy. But I am here talking to all of you. And I bring that up because I think that's very empowering for people like me. And I didn't used to do that. And I started to do it, since the George Floyd situation. I started talking about it more and more. And I see the difference when I talk to people that are not the traditional scientists. So there is room for you here, and I'm proof of that. So follow your dreams, and just talk to us.
Thanks a lot to both of you. Let's jump to the next concept, ecosystem. This time illustrated by the Rocky Mountains, Lander Peak, artwork by Albert Bjørstad. So we are going back in time. And I would like to hear a little bit more about something you have already mentioned, Thomas, in your presentation, and when you illustrated it with the Brucker and Bartenstein 2013 figure, right, where the holobiont ends. We have the host, we have the associated microorganisms, which are part of ecosystem, and we can see in that figure. So there are many holobionts in this ecosystem. And so my first question would be probably easy to ask, probably difficult to answer. Where does a holobiont end?
I answer you with another question. What is an ecosystem? You are an ecosystem. And maybe a better way of talking is about nested ecosystems. If you say an individual or a holobiont is an ecosystem, this holobiont is embedded in many other holobionts. And so at the end, it doesn't really end anywhere. I mean, it goes, and I think if you think that further, then I showed you that review summary, this graphical abstract from that review here, then you end up with this aquatic terrestrial ecosystem unity, holistic understanding. And I mean, so many people talk about One Health these days, but that's probably just the essence of that is that everything is connected to each other. And I mean, entangled banks, I mean, including the invisible ones. So I think there are no boundaries. And the wonderful graphical abstract by Robert Brugger and Seth Bodenstein they draw a line between the hologenome and the environment. But as I tried to say, this line is perforated, and so that bacteria or microbes or symbionts, or potential symbionts, can come in and others can go out. And so there is no strict boundary. And maybe that's the secret of life, how to adapt and how to stay stable. Yeah.
Mm-hmm. And Monica, how would you say that this fuzziness, let's say, that this permeability translates into actual experiments? Because in the experiments, you really need to design something. You need to set some boundaries somewhere. What are your thoughts about this? How can we find a balance between being permeable, but then being able to experiment and set a hypothesis and test whether this is correct or not?
I think that you have to first sit down and think of your technological challenges. Like, I can't do what Thomas does with Hydra, right? So going in with that hard understanding of what it is that you can do with your system is really critical. And then make sure that you define, if you think that this boundary of, for instance, loosely associated microbes, it's important to include in your holobiont, and then those transient microbes as part of the ecosystem for the specific question you're asking, then you need to be explicit about that, because then the critics will have a harder time. And just defining this is for this purpose to answering this question. These are the boundaries that I'm setting on my system. And we've had to do that when I started thinking about photosymbiosis. I was starting to think exclusively about the algae, and soon after that I was like, I can't just think about the algae. I can't. But that adds that level of complexity that is overwhelming, because it's a complexity that you don't even know. You just know it's there. So how do you partition that complexity? I think it has to come from your experimental design and immediate question. And then you can redefine it, right? Like, for instance, in coral biology, people have started to put different boundaries at what is in the water column, because there is this layer of microbes that are attracted to the coral that are more frequent visitors. And it's microns or millimeters in length. So there is this gradient of microbial assemblages from the mucus of a host towards what is in the environment, and that's the same thing probably in the rhizosphere, right? What is right inside, the legume, rhizobium interaction versus what is on the surface of the root, what is in the proximity and farther out, or so I think trying to define those boundaries and maybe excluding things in your immediate quest is important too. So I don't think that we can any longer be as sticklers to the definitions of the past, and just make sure that you define the concept that you're using as you move forward. Like your concept of microbial biogeography is the first time I hear of that, so I need to ponder. It's like, trained as an organismal biology, I think of biogeography very differently. So, okay, I'm gonna pause and think about this. So as long as you be upfront of this is how I'm defining this word for this purpose, we can gain a lot of knowledge through that. It's just being clear in how you're using these words that have been given to us by previous scientists.
Mm-hmm. So if I can add here one last statement or opinion of mine. We talk about different models, and you work on different models, and each model has, and I told you, a model is a lie, but which helps you to understand the truth. But I think, as Mónica was alluding to, it's important which question to ask to what type of model. And the worst thing is that you ask whatever, a cell cycle regulation, mutant question, which you can easily do with genetics and yeast, now with a non-model system. So you think about, that is the limitation then of understanding. Think about which question is most appropriate for which type of model, and then you will succeed. And corals are different from Hydra, but there is an environmental, there is an economical importance, there is more complexity, and so each model has, in terms of career advisement, I always use this word, unique selling point. What's the unique selling point for your model? And that you have to play, that card you have to play, and then you get your money. Or your Maserati, whatever.
We have actually a question from a person in the audience that does work on microbial biogeography. And his question is related to what you said about the transient bacteria, resident bacteria. So he's asking, to what extent is the microbiome truly selected by the host versus assembled independently? How much do we know about that? And does it differ a lot across different host systems? Mm-hmm. Or are there any universal rules we can already tell about to the audience?
Well, so be careful. And I think we have good examples. The question is how stochastic is the colonization, and how specific is the colonization. So we have good examples where we clearly know, and I refer to Seth Bodenstein, and he has written a number of summaries on reviews on that, where we have what Seth called phylosymbiosis. And if you have a phylosymbiotic relationship, then there is a selective process involved where the host selects for a given microbiome, and that is stable over evolutionary times long. So that is not very stochastic. That is selected. But I would not exclude that there are organisms or organs where there is much more flexible term of colonization. And when I talk with Margaret McFall, we always talk about tourists and residents. There's tourists coming to Copehagen, and so we all disappear on Wednesday again, but there are residents here. And I think the important guys are the residents, and but tourists may also add an additional added value. So I would say there is no black-white. Yeah, there is a certain type of environmental input, and microbes can settle and colonize from the environment, no doubt. Other term which is used in the field is the so-called core microbiome, to distinguish that core microbiome, which is then very stable with a given host, from the more environmental, habitat, nutrition, whatever physical factors dependent microbiome. So we talk about a complex system, so you have to accept complexity, and there is no yeah, it's not the one side or the other side. There is in between, but that's the power. And just go back to nature. That's the power of nature, of organisms, of evolution. They are resilient because of these gray zones, which are making them obviously so resilient and successful.
I'd like to add to that, just hearing you talk about torus and resins reminded me of, you know, these great thinkers in the origin of life, like Jim Lake was such a huge proponent of horizontal gene transfer in origin of life. And if you look at history, it's a ring of life. It's not, a branching tree. And then recently, Ford Doolittle published this idea that it is Knott's hypothesis in PNAS. It's an acronym for it is the song, not the singer.
Yeah, it's the song and not the
right?
singer.
So there is so much redundancy in microbial communities that maybe a host just picks up not its favorite microbe, but the one that does what it needs right then, So there is that functional redundancy that then throws that whole new level of complexity of how you're approaching your question if you're just looking at function rather than species boundaries, which are very fuzzy when it comes to microbes. So I think that it really depends on the question you're asking, because if you want to use that for biogeography, it can get really messy really fast, right? And then if you think of, like the mosaic, coevolutionary mosaics in the context of biogeography, symbionts in a northern range are going to be very different from microbial symbionts in a southern range or things like that, and there is evidence for that. So the biogeography of the microbes will be different from the biogeography of the host. The host could have a lot of gene flow, but there is environmental parameters that are selecting for high latitude versus low latitude traits that the microbes are the ones that are contributing to the biogeographic patterns. So I don't know if I'm answering the question, but that's my 2 cents.
Is there anything else you would like to add?
I just want to support what Mónica just said. I think this concept for Doolittle, it's a song and not the singer, is really, many of us for a long time thought that if we have identified a specific bacterium or a specific microbe in a given location, then this is the guy who is important. But it's actually not the species per se, it's the physiological output, the metabolic output, and that can be done in a different organism. That's why every individuum here has different microbiomes, but different microbes can have a similar metabolic output. So all your sequencing will not help, and this is why we still do not know what is a healthy microbiome, because different microbes can do the same thing. So yes, important. Liping Chow talks about physiological guilds of microbes. So a guild is defined as a functional output unit, and these guilds can be composed of different members. And so yeah, we are living in a kind of complex world, but it's so fascinating.
It is, indeed. And I know we will have many talks that will touch upon all these concepts that you are mentioning, so we will have a chance to discuss further in the next 2 days. But now, next concept, which is challenges. And Thomas, in your talk, you mentioned in the last 9 points that you listed there, the value of the Holobiont hologenomic view to address also challenges, global challenges that we are facing now, not just the humanity, but also at the planetary level that transcends the human in themselves. What would you say is the main advantage that we get by taking the holo perspective compared to the reductionist perspective that you were mentioning towards addressing these challenges and coming up with new strategies to address those and hopefully save our future?
It's very straightforward and a short answer. So I'm also a member of the medical faculty in my institution, and I'm very happy and glad that I'm surrounded by medical doctors who are convinced, and there are not all medical doctors convinced, that if you have a skin disease, a neurodermatitis, for example, or a gut disease, inflammatory bowel disease, or a metabolic disease like obesity, or a neurodegenerative disease, that you will never understand that if you look at the skin epithelium, or in the intestinal goblet cells, or the Punnett cells, or in the neuronal network alone. The challenge and the future of medicine is a holistic medicine, and few people really say that. We have a chair in our institution of evolutionary medicine, but good and I would say wise people medical doctors are accepting that. So this is challenging, but there is no other way to understand this complex diseases, which are now lifestyle diseases, and where millions, 60% of Germans and probably the Danish are all living very healthy, of German American people are obese. There is an ever-increasing cases of allergies, of inflammatory bowel diseases, of neurodegenerative diseases. And it's not that because we all live much longer. I always get this argument, this is stupid. Many of these diseases are just affecting young people. It's lifestyle. And the only way, and there's no end of the trend yet, which is horrifying. I think the whole science should actually concentrate on that issue, that every nation has millions and millions of suffering people, and it costs millions and millions of Danish krones or euros or dollars. And a lot of people make a lot of money because of that diseases, but we should start to treat them and to see them as holistic problem. And holistic, I mean very simple, as holobionts. A patient is a holobiont. And then you can start to get to the ground of the problem, to the base of the problem. And there are institutions who do that, so-called comprehensive centers, where a patient is seen by not only the expert of the skin or the intestine, but also by a sequencing guy and others, and modelers. So you have to, I repeat myself, you have to embrace complexity to solve these issues. Is that
one question about, related to complexity. So you also mentioned before that we need to make use of these very advanced technologies. And those advanced technologies are not reachable for everyone in this planet. At the same time, we have challenges that are applied to the entire planet, and sometimes also half of the planet that is creating challenges for the other half of the planet. Monica, you mentioned that coming from Colombia, that perspectives can be very, very different. And how would you say that how can we ensure access for researchers from non-rich countries so that they can actually take advantage of this holistic view, get access to these technologies that enable us to go to that reductionist interactions, but considering the huge scope of the complex holobionts. What's your view of this? Is there anything we can do from Denmark, for instance, so that all this research is not centralized in the wealthiest countries?
Okay, I have several things to say. I'll try to remember them all. I was at 3. I'll start by answering your question. I think that there is movement in the right direction. I think I wouldn't say any longer it's too little too late, because I am hopeful. I'm trying to be very hopeful. And I think that it takes a village. I see a lot of positive movement in the right direction, finally, beyond performative from the journals. And I think that is great that the journals are actually, because when I edit for a journal, I always ask them these questions, now. And I see their actions, and I think there's a lot of things happening behind the scenes. they're coming up with good ideas of how to open the platforms beyond we'll waive your fees. I think it needs to go beyond that to encourage submissions, to encourage design. So the journals are one thing, the funders are another thing. For instance, the foundations that fund conferences like this. I just sent a bunch of people in Latin America this announcement by the Company of Biologists that are starting to sponsor meetings in the Global South. And those are such great workshops. I attended one of them, and it was really, really, I think that's the last time I saw you, right? In that castle in England. Yeah. Amazing, right? Why not do this in the Amazon, right? In the desert in Africa, and bring those local scientists and blend those great minds together and just elevate those people and bring people down that want to help. The conferences, multiple funding strategies that, I mean, a little bit of money can go such a long way in a developing country, such a long way, because it's cheaper for certain things, certainly access to all these endangered ecosystems, right? To the health challenges that indigenous communities are suffering in Amazon. Now, Starlink is putting cell phones to all these indigenous communities in the Amazon, have cell phones, collect data that way, you know, so we can help them with diseases and stuff like that. So I think technology can be really powerful, but I think it's just pouring your heart into it, and I see positive movements in the right direction. I think there's got to be a lot more funding that is multi-nation, but elevating the Global South scientists as partners and not colonial approaches of, we go collect the poop from the rhino and bring it back to Copehagen and do the microbiome analysis, but bring the scientists or bring the science there, bring the Oxford nanopore machine and train everybody in doing that. It's a pocket instrument now with the laptop. Things like that, I think, would be revolutionary. So that's my question to your answer, but I think another challenge for us in this room that is really important, I see this community as what is the best word? The guardians of holobiome research. I think that the term can lend itself to so many things that we need to be careful. It's being hijacked by dubious characters, companies with questionable goals using the term. I saw a chart presented by a philosopher of science many years ago now, where it went from that debate among people like us, and the evolutionary biologists going after Seth and saying, and just being a conversation among intellectuals to these things that people want to use for profit. So that is where we, as the reputable scientists working in holobiome research, need to be very careful that we have set a steady course and standards. And I think that being, more focused is a big challenge that is on all of us to be careful moving forward, because there is thousands and thousands of citations that have no business, or companies using the term holobiome or hologenome out there that will undo what we're trying to do here. So that is an immediate challenge. And then the third one, I know I'm being really long, I'll be fast, is to go back to something that we've already said, that I think there is more openness towards using microbial approaches to address the health crisis, the ecosystem crisis. In corals, we're so desperate. It's a matter of years now, really. And what Raquel and others are doing with probiotic approaches it's in its infancy, but they're champions, and they need to be supported, and more people need to join to help with using that component of the holobiome that is going to help us reverse the damage. So that's a big challenge.
Mm-hmm. Thank you, Monica. So we have Tom's shadow already here, which means that we need to come into an end. So I would like to ask you, the two of you, just a short statement for the audience. There are a lot of young researchers. You have already touched upon some of the important points for the young researchers. But what would be your message for them? Do you have? Yeah. So we're going to start with Thomas, and then we will end with Mónica.
To the young generation, the future is yours. I mean, I mean it really. That this field is, I wouldn't say exploding, but it's just a new field which is now kind of on solid grounds. And develop that further, find your niche and find your questions. And it's not only intellectually fascinating, but it's also extremely important to move along on that. And you will find a job and you will find fun.
And Mónica, what would be your last words? No more for this conversation.
Embrace diversity. It's a big one. Embrace diversity, because, you know, people that you think, or your ideas that you think are on the fringe of something may bring that new breakthrough, and we need a lot of breakthroughs today to turn things around for the world and for science, and stick with it. Look, you know, aim for the long haul. Look far away, not the immediate, because it's hard. Funding is getting more complicated. Science is getting better. So that's the upside of not being funded every time you try. That means the quality is so good that we're all doing something good collectively. So of course it's going to be more challenging. So don't just get discouraged by the immediate hurdles, but, you know, keep looking towards that farther away goal. And if you see an opportunity to pivot and jump into something new, take it. Go for it.
Thank you. So my last words will be for the audience. My apologies for not being able to add more comments and questions into the conversation. I am pretty sure that a lot of people, everyone here, will be really keen on discussing about these topics, about these questions that you have posted in Slido. So thank you so much for this wonderful conversation we have had, and I really hope this will continue and we will be able to have a really constructive conversation in the next 2 days. So now we have the reception. So